Good morning, everyone, and thank you for joining us today. My name is Katy Stubbs, and I lead the public engagement team here at Alzheimers Research UK. It's my pleasure to welcome you to the 7th event in our Lab Notes online series. Today, we're hearing from researchers who study important housekeeping processes in the brain that make, fold, and dispose of proteins, and how these processes can go wrong in dementia. This event is being recorded and will be added to our website and YouTube channel early next week. If you've missed previous events, you can watch any of them back in your own time. If you would like to use subtitles during this event, you can turn them on using the CC button at the bottom of your Zoom window. The subtitles are auto-generated live and so may contain a few mistakes, but all of these will be edited and corrected on the recording. So before we get going, we've got a couple of poll questions for you, and they should pop up on your screen any second. And these questions help us to understand your reasons for attending today, understand your current level of knowledge about dementia, and whether you've attended one of our events in the past. So I'll pause very briefly just to let you have a few moments to answer those. Okay, so hopefully it's given you a bit of time to pop your answers in there. So if we could see the results of that, that would be great at this point. There we go. So people rating their knowledge about average coming into the events, and people coming along because they have some personal experience of the condition, or their work is related to it, or they just actually want to find out more about research. And we've got a good mix of people who have come along before and a few people who are coming along for the first time. So it's wonderful to have you. Thank you so much for joining us. So let me just get going and share just a couple of slides as part of my introduction. So just a few minutes to hear from me about some updates from us at Alzheimer's Research UK, before you'll then hear two talks, one from Professor Jody Mason and the second from Professor Myra Conway. We'll then move to the Q&A session, and we'll try to answer as many questions as possible in the time that we have. Many of you have submitted questions in advance, and you can also ask questions during the event now. To do so, just click the Q&A button at the bottom of your screen, which will bring up a box where you can type your question in. So first, I just want to tell you who we are in case you're coming along for the first time. So here at Alzheimer's Research UK, our vision is a world free from the fear, harm, and heartbreak of dementia. And to deliver this vision, we fund biomedical research, supporting scientists working to build our understanding of the biology of the diseases that cause dementia. And this research is helping to find new and improved ways to reduce risk, make earlier and more accurate diagnoses, and create effective dementia treatments. So our policy team keep a watching brief on what's happening in the sector of dementia research. And last week, they released a new report about clinical trials for dementia. As you might know, clinical trials are vital in the development of new treatments, but we know there are a number of challenges around them that make it harder to deliver much needed treatments for dementia. So the report has a number of takeaways, which I'm not going to go into. I just wanted to highlight a couple. So one key challenge when running a clinical trial anyway is recruiting volunteers to take part. For dementia trials, we often need people who have very recently been diagnosed with dementia and so are at the milder stages. However, our report has found that only a very small proportion of people are told about opportunities to take part in research at the moment that they are diagnosed. This means it can take years to recruit the numbers of people needed for these studies. So one key thing we need to do is to better embed these research opportunities and schemes that we run, like joint dementia research, into the diagnostic process. Another really key thing we need to do is to engage better with communities that have traditionally been underrepresented in research. This will ensure that studies have better diversity in the volunteers taking part and that their findings are more widely applicable. As I said, there are lots of other recommendations we make in our reports to improve the clinical trials landscape. You can find out more about this report and the outputs from it on our website, and we've put the link in the chat in case you're interested in looking at that. And the second bit of news for me this week is a really lovely fundraising story. So our researchers in our Scotland network marked Dementia Action Week last week with an amazing fundraising effort. A team of 20 dementia researchers ran, walked, cycled and swam 1,193 miles over 7 days. And together they raised £3,410 for Alzheimer's Research UK. They originally set out to cover about 500 miles, roughly the distance between the different universities in their network. But having hit their mileage and fundraising targets midweek, they decided to double them. So it was wonderful to see their passion for dementia research go the extra mile with their fundraising efforts. So I just wanted to share that and say a huge thank you to them for all what they did last week. So this Lab Notes event and others in our series are brought to you by us along with our research network. And we provide funding to our network to support small projects and pieces of equipment to help scientists network and collaborate with each other. Before the pandemic, each centre would organise an annual event to share their work with people in their local area. And so we've worked with them to bring these events online. At this event today, you'll hear from two scientists in our Bristol and Bath research network. Professor Jody Mason leads a research group at the University of Bath, and Professor Myra Conway does the same at the University of West of England in Bristol. They will both speak about key housekeeping processes that go wrong in Alzheimer's disease and why proteins and the making, folding and disposing of them are so important in dementia. After they've spoken, I'll come back on screen and we'll have the Q&A session. So without further ado, it's my pleasure to introduce Professor Jody Mason and get started with our first talk. Welcome. Thank you, Jody, and it's over to you.
Thank you very much. Thank you very much for that kind introduction, Katie, and for the opportunity to speak. I'm just going to put myself into full screen mode. Can you see my screen? Hopefully you can. Fantastic. Okay, I think we're away. I just need to switch on my laser pointer. Okay. Great. So what I would like to speak about, I guess, for the next 15 minutes or so is the fact that at its core, Alzheimer's disease is a protein misfolding disease. So I'll go into detail into what that means, but you can see here at the bottom sort of left-hand side that we have a highly regular amyloid fiber here. So this is a regular repeat of protein which is clumped together into things that we call fibers, and it's really that process that's happening in the brains of people with Alzheimer's disease. And to really go back on what I mean by that is really to go back to Alois Alzheimer and his first diagnosis. So he was obviously the first person to really show that Alzheimer's disease is a disease. It has an underlying pathology. And in 1901, he came across this lady here, Augusta Dieter. She was 51 years old, and she'd been admitted to an asylum in Frankfurt with behavioral problems, severe memory loss. And what Alois Alzheimer did was to really keep a document, a diary of her condition as the Alzheimer's disease progressed. And he did that for about 5 years. And then when Augusta passed away in 1906, Alois was able to then take her brain from Frankfurt down to Munich. And there he was able to access a laboratory that could utilize a new type of stain. And by comparing from brain slices, the brain of Augusta Dieter to people that had passed away of natural causes, he could see these inclusions, which he called plaques and tangles. Now, you know, we now know that these things are protein deposits. So plaques are predominantly made up of a protein called beta amyloid. It's a very small protein. In fact, it would be called a peptide. It's so small, so it's only about 40 or 42 amino acids long. And then inside the brain cells, we see the aggregation of a much bigger protein called tau, which forms these things called tangles. Okay, so does it affect only older people? Well, it depends on how you define old. It affects about 1 in 20 of us over the age of 65. So I think we'd all agree that 65 is not an old age. In fact, you're not even retired at 65, you know, these days. And so 1 in 20 over the age of 65, and that obviously increases as we get older. So it is an age-related disease. As we get older, the chances of these misfolding events increase, and that can lead to an increased risk for Alzheimer's disease. Although incredibly rare, it can also affect younger people. So this is certainly less than 5% of all cases. And we refer to this as early onset Alzheimer's disease or familial AD, where there is a genetic sort of predisposition. There's something that sort of underpins this. And that's taught us much of what we know about the disease, because it really points the finger at key genes and at key proteins and key players in the process, and a lot of that then points the finger directly or indirectly at this protein called beta amyloid. And again, as I highlighted on my previous slide, Alzheimer's disease is not a normal part of aging. As we get older, it's normal for our brain to not be quite as sharp as it was, but there is this underlying pathology, as Alois Alzheimer saw over 115 years ago now. And so it's, yeah, as I said, it's not a normal part of aging, and it's normal as you get older to get a little less, you know, a bit more forgetful, but it would not be normal, for example, to go out and to not be able to find your way home. So to talk about misfolding, it's helpful, I think, to go first back to the concept of protein folding. And to do that, we need to think about the DNA, of course. The DNA that we all have is our genetic blueprint that makes us who we are. And one of the things that DNA does, or the genes within the DNA, is tell cells how they should make proteins. So the proteins are life's workhorses, and they really carry out the instructions that have been encoded within the DNA. So these things can be enzymes that help to catalyze biological reactions on meaningful timescales. They can be structural proteins, such as keratin that you find in your hair or in your nails, that have these structural roles. Or they can be functional role proteins, things like hemoglobin, that has to be able to fold up into a very unique three-dimensional structure so that it can bind to oxygen in our lungs and then distribute it throughout the body wherever it's needed. And so proteins are basically, in the simplest form, are just beads on a string, where these beads are amino acids, and it's the number of amino acids and the combination of amino acids that dictates the protein and how it will fold. So at some point it goes from an unfolded structure, just beads on a string. You can think of it as a sort of floppy piece of spaghetti when it's just been produced, and it folds into this highly unique three-dimensional structure that it needs to be able to function. And most of the time that's fine, there's no trouble at all, but very occasionally what can happen is proteins can misfold. So at some point they go from this floppy piece of spaghetti, the unfolded state, and they don't make their way to this highly defined three-dimensional structure, the folded state. They go off the pathway. So you'll see here this term I, this is an intermediate within the folding pathway. Something causes this protein, instead of making it to the folded state, to go off the pathway. And when it can do that, you see these arrows, these can stack rather like plates and growing into these long amyloid fibers that I showed you on the very first slide. And as I said previously, Alzheimer's disease is an age-related disease. So as we get older, our chance of having Alzheimer's disease increases, and it's part of a subset of other age-related diseases. Other examples including things like prion proteins and CJD, type 2 diabetes, there's a protein called amylin, Huntington protein, alpha-synuclein in Parkinson's, and there are about 20 others or so. So there's an underlying pathology to all of these diseases, and all of these involve the misfolding of proteins into amyloid. So in Alzheimer's disease, we're talking specifically about this protein beta amyloid that stacks and grows into these long fibers. And more recently, we've been able to move away from these rather sort of crude images that come from electron microscopy, and things like solid state NMR and cryo-electron microscopy have allowed us to drill down into much greater resolution into these structures. And you can see here the resolution is very good, and that allows us to see individual amino acids and how they're organized. We can see here, by turning this 90 degrees, we can look down the axis of the fiber, and you can see this S-shaped fold. It's referred to as a Greek key. And we can also see that there's rotational symmetry around the axis of the fiber as well. So having this high resolution structure really tells us a lot more about the disease, and it helps us as well to think about therapeutics and how we might want to target these proteins and sort of think about how drugs might be able to bind to them. And one of the things about beta amyloid as well is that it can grow into these very long fibers, but it can also populate a range of very smaller, sort of shorter, more soluble types of aggregates. So they don't always have to be these long forms. And it's in fact thought that some of these shorter forms can be the most toxic forms of the protein. So what's all this doing to the brain? Well, in essence, it's leading to loss of brain cells, of course. It's killing the brain cells. That's leading to the symptoms of the disease. And you can actually see a loss in the overall mass of the brain as these cells are lost. And Alzheimer's Research UK had a campaign, I think 1 or 2 years ago, highlighting really the fact that you can lose up to around 140 grams of brain mass, which equates to about the weight of an orange. So again, this underpins the fact that this is a physical disease. So again, here looking at the bottom, we have a positron emission tomography scan. So this follows brain activity using a radio tracer. In this case, it's fluorodeoxyglucose. And you can see that the decrease in metabolism in an Alzheimer's disease brain as those brain cells are lost. So how does this come about? Well, the beta amyloid protein that I showed you in these long fibers, in these amyloid plaques, again, that Alois Alzheimer saw when he looks down with his microscope, this is actually chopped out of a much, much bigger protein. So this protein here is called amyloid precursor protein, or APP for short. And you can see the bulk of the protein is found outside of the brain cells, but part of it passes through the cell membrane, the cell wall, and inside the brain cells. And something causes enzymes, or small molecular scissors, if you like, to come along and snip this protein. And they snip out a very small section, which is the actual beta amyloid protein. Now this can vary in the length, so it could be 40 amino acids, or it could be 42 amino acids long. It could be a range of other sizes in that range. And when these enzymes or scissors snip this part of the protein out, it's that then has the propensity to start to self-associate, so start to stick to other molecules of itself and clump into these amyloid plaques that are known to be toxic to the brain. So if we zoom in on that yellow section then, and we look at the scissors around it, we look at the changes, you'll see that in early onset cases of Alzheimer's disease, you'll see all of these individual amino acid changes, and you'll see the location where they were first identified and how that points the finger to this protein and these particular enzymes as well. So for example, we can see the Swedish mutation here. So a couple of amino acid changes close to this pair of scissors means that it's cut more readily. So the analogy I would give is the scissors obviously in the piece of paper. So if we have changes in the amino acid around this site here, it makes the paper easier to cut. Or we can actually have mutations within the scissors that mean that they're sharper. So these pair of scissors, for example, are called beta secretase. These pair of scissors are called gamma secretase. So these can become more sharper in early onset cases and cause the scissors to either cut more often, or they can cut in slightly different places. So they can lean towards this extra 2 amino acids, which is A beta 1 to 42, which has a higher propensity to misfold and become sticky. You'll also see that there are changes actually within the yellow section. So these don't change the rate at which this protein, this APP protein, is cut, but they increase the chance when it's been produced of then misfolding and clumping and sticking together. So I've highlighted some of the evidence here for A beta in Alzheimer's disease. We know that it can aggregate into toxic fibers and plaques. I've highlighted the fact that actually it could be some of these smaller, more soluble types of A beta clumps that are more toxic. These plaques are often used to actually diagnose Alzheimer's disease. Mutations within that bigger protein APP can lead to early onset AD. Mutations within the scissors, the beta and the gamma secretase enzymes, can also lead to early onset AD. And then we can have mutations within APP that can increase the amount of the 42 amino acid version that's produced, or perhaps the ratio of the 42 over the 40. And if we build some of these genetic changes into mice to create transgenic mice, they also develop Alzheimer's disease-like symptoms. So that's obviously very compelling evidence. And finally, people with Down syndrome who have 3 copies of chromosome 21, where the APP gene is located, will typically develop amyloid plaques within their 30s, and some Alzheimer's disease-like symptoms will follow as well. So what can be done to try and prevent Alzheimer's disease? What are some of the approaches being leveled at this disease? One actually would be to prevent the amyloid precursor protein from being produced in the first place. Obviously, we go from DNA. I said that's the genetic instructions that code for this protein. It passes through this intermediate RNA, and we can try and create things that would interfere with the RNA that's produced. So maybe small interfering RNAs that can then block the production of the APP protein. Or we could try to blunt these scissors. So we could prevent the APP protein, this much bigger protein, it's 700 or 800 amino acids long. We can blunt the scissors, particularly the beta secretase and the gamma secretase, so that it doesn't get cut and this yellow section doesn't pop out. There's actually a third pair of scissors which can cut in the middle. This is alpha secretase. And if we could allow these scissors to cut, or perhaps we could sharpen them and increase the activity of this particular enzyme, then perhaps we could cut the protein in the middle, and that would be an alternative way of solving the problem. And there's work underway looking at all these different approaches. The third way that we could try to level against beta amyloid, and this is an approach that my group here are taking at Bath, is to allow the protein to be produced, allow the scissors to cut so that A beta is produced, but once it is produced, to stop it from then clumping so that it can stack like plates into these long amyloid fibers. And so the way in which we're doing that is we're screening peptides, incredibly short proteins, maybe just 5 or 10 amino acids long, and we're screening many millions of different peptides, different combinations of amino acids that are strung together inside living cells, and then we're throwing these against the A beta that's produced, and we're looking at those that can stick, but more importantly, those that can detoxify the protein. We know that when it starts to stick, it becomes highly toxic, and we know that that toxicity then kills the cells. So if we can screen our peptides by finding those that can stick, that can detoxify the protein, and of course, because it's done inside living cells, ultimately anything that is toxic will kill the cells. So we have to, we know that it's a good readout when our peptides combined and the cells are happy and can express all of these proteins, then that's a very good sign. So there's reason to be optimistic, and the outlook, I think, is good. I think there's a lot of work left to be done. I've highlighted one particular protein that is very predominant in Alzheimer's disease. I mentioned a little bit tau as well, which is another protein that is a source of a lot of academic and industrial inquiry as well into Alzheimer's disease. One thing we do need to look at is the fact that the pathology is occurring probably several decades before the symptoms start to develop. So another piece of work that's underway is to develop things called biomarkers of Alzheimer's disease that can start to inform on the pathology that's going on before the symptoms develop. So if we can develop biomarkers and we can develop drugs, then we can get our drugs in earlier before the symptoms are starting to present, and then we can have a good chance of combating the disease. So coupling these two together could be one key thing. And it's worth noting that even a very poor drug, a weak binder that has a low efficacy, could have a positive outcome on the disease. And it's worth knowing that if we could slow it down, slow Alzheimer's disease progression down by 5 years, it would already cut deaths by approximately 50%. So I'm going to stop there, and I'm going to now hand over to Professor Myra Conway. Myra is a professor of biomedical sciences at the University West of England, and she's going to be talking about autophagy and its role in Alzheimer's disease. And this is an important clearance pathway for the removal of proteins. So I shall hand over to you now, Myra.
So good morning, everyone, and thanks very much to Jody for introducing me. I'm now going to share my screen with you, and we can get started on the talk. So here we go. So thank you very much to Jody for introducing me with respect to my talk on autophagy and Alzheimer's disease. And this really is an example of a process or a mechanism in the cell that happens downstream of protein misfolding or aggregates. So these type of buildups or components that happen within the cell can stimulate autophagy, and this whole mechanism is thought to be dysregulated in Alzheimer's disease. And today my talk will focus on what autophagy is and how this can be controlled and to try to regulate it under disease conditions. So first of all, what is autophagy? So in a healthy cell, we can maintain that healthy cell through the process of autophagy. So as one can see here, when there's aggregates built up in the cell, or organelles that are not working or effectively, or there's debris in the cell, what happens is autophagy forms a membrane around those vehicles, which eventually develop into what's called an autophagosome. And this fuses with a lysosome that contains chemicals that will digest this material. So effectively, autophagy is like a recycling system or a manner by which debris can be removed. Now, this is different to cell death. Now, in cell death, the cell can no longer cope with a recycling system to maintain activity, and it signals the cell to die. And here you get the nucleus, which is starting to decrease in size. You're going to get this blebbing, which is the pulling off of material from the cell, and the cell shrinks over time and eventually causes cell death. And I liken this to, we'll say, for example, when you start off having a brand new car, it's really fantastic, it's functioning fantastically. However, with time and as the car ages, so just like the cell, as the cell ages, that car may not be operating or functioning as well as before. Now, there can be cases where the car is irreparable and that needs to go on the scrap heap. So the car dies. And that's the exact same thing that happens with the cell. The cell can die. Now, this is unlike autophagy, whereby a healthy cell, again, over time starts to become a bit dysfunctional. It's part of the cell is not working properly, part of the organelles are not effective. And just like a car, your engine might break down, your handbrake may not work. All of those parts of the car may not be working. But if you send it to the mechanic, they can repair this car, and then effectively your car is as good as new. So that's just like autophagy, whereby the cell has this recycling system to make it just as good as new. So in essence, this process within the cell optimizes cell function and maintains homeostasis. It retains the beneficial substances, such as nutrients, for example, and that can help a cell to survive under conditions where there's nutrient deprivation. Importantly, from the concept of Alzheimer's disease, it removes harmful substances. It can eliminate misfolded proteins or aggregated proteins. So those proteins that Jody was talking about that build up in the cell, autophagy is a process that removes or gets rid of this. So in essence, it's out with the old and in with the new. It's like a whole refurb of the cell, remaining or maintaining, I should say, functionality. Okay, so it's really important for us to understand how autophagy is regulated. Now, when we're talking about diet, we often think about, your cardiovascular risk, the importance to your liver, for example. But what's more important in the context of Alzheimer's disease is how that gut-brain axis works. So we very much need to think about what we're taking in our diet and how this can improve or maintain a healthy brain. So, for example, today we're going to have a sandwich, and that will contain carbohydrate, proteins, and fats. And we're going to concentrate on the protein element. So as we break down proteins, these are broken down into amino acids. Now, the amino acids, there are 20 amino acids, 9 of which are essential, i.e. you've got to take them in your diet for them to be effective. And just like Jody says, these amino acids will line up to make a sequence or a code, which looks like a string of beads, and that will tell the cell what type of protein it needs to make. So amino acids are the precursor for protein synthesis. So under fed conditions, the body will use that dietary protein to produce amino acids such as leucine, and leucine is one of the main nutrient signals, to create new proteins. So it's important in protein synthesis. Now, conversely, under fasting conditions, under our nutrient deprived conditions, if your intake of protein is very low, then in that particular context, you won't have the precursors, the substances for protein synthesis. So therefore, fasting causes a stop to protein synthesis, but then it stimulates autophagy. And the reason it stimulates autophagy is because the cell still needs nutrients. And autophagy is a way of recycling these materials and giving the body nutrients. However, it is not just diet that controls this mechanism. Also, autophagy can be stimulated or affected by the buildup of misfolded proteins, damaged organelles, or aggregates. So now we've got a bit of knowledge as to how autophagy is regulated, and importantly, how the purpose of it. So if it's such a good thing, why are we bothered to study it in Alzheimer's disease? So in this particular slide, we can see that autophagosomes are accumulating. So what they've observed is that in AD brain, these autophagosomes start to build up. And just like I showed you at the start of the talk, what can happen is that they believe, or our research has shown, that there's either a problem with the synthesis of the autophagosome or in the clearance of the phagosome. So ultimately, some part of these pathways has become dysregulated. And as a consequence of that, there's a buildup of aggregation or aggregates within the cell that would normally be cleared. So this is why we're interested in studying autophagy in Alzheimer's disease. So just a little bit about our research. So I'm interested in two proteins called the branched chain aminotransferase proteins. In this talk, more specifically, the cytosolic form, which is found predominantly in the brain, and the mitochondrial form, which is more ubiquitous, is found everywhere in the body. I'm interested in these proteins because fundamentally they play a central role in breaking down your leucine, isoleucine, and valine, so your branched chain amino acids, into their component parts that is important for energy. And a lot of work has been done about its role in protein synthesis, but nothing or very little is understood about, or if any, does it have a role in autophagy. Now, we showed previously in our work that the BCAT proteins are significantly increased in AD brain relative to the control brain. And importantly, this correlated with BRAC stage. So as the condition of BRAC or of the disease progresses and increases in intensity, the level of the BCAT protein also increases, indicating that as the severity of the disease gets worse, the level of the protein is really high. So this can mean it's a consequence of the disease, i.e. it happens as a result of the disease, or it can play a role in the pathology. So if we go back to our nutrient-fed conditions, so under nutrient-fed conditions, if you recall, I mentioned dietary proteins give rise to amino acids, give rise to protein synthesis. And given the role of BCAT C in this mechanism, we considered that if we fed our cells, we'd have a lot of BCAT C in there. And in our first little picture here, we can see that there's some BCAT C, but is not as much as the marker of protein synthesis, which is shown in red here. Conversely, when we nutrient deprived or starved ourselves or fasted ourselves, we see that the level of the protein increases quite a lot, indicating that rather than BCAT C playing a fundamental role in synthesis, it may be just as important or more important in autophagy. So to look at it this further, we took our fasting cells, and what we showed is that the cytosolic protein here, BCAT C, is indeed increased under these conditions, and it also aligns with the increase in VPS 34, which is a marker of autophagy. When you combine these two images together, you can see a lot of yellow within this particular image, and this indicates that the two proteins are interacting. Just here as another little bit of information, rapamycin is currently under trial because it's important in inducing autophagy in models. So they believe that by inducing autophagy, can they help to promote the clearance of aggregates? And what you can see is that rapamycin also causes an increase in BCAT C, which is directly aligned with the VPS 34, a marker of autophagy. So this evidences that BCAT is really important in regulating the autophagy mechanism. So I liken my theory of this particular part of the talk to the Goldilocks theory, whereby too much of a protein or too little of a protein is really not great for a cell to function optimally. So the balance has got to be just right. So if I take out BCAT from my cells, what happens is that my marker of autophagy, or my markers of autophagy, significantly decrease, indicating that low levels of BCAT correlate with low levels of autophagy. So that's really important in maximizing the efficiency. Conversely, if I have high levels of BCAT present in my cells, which is what we see in AD brain, this can be related to an increase in autophagy markers, as we can see here. Now, this indicates that autophagy is really upregulated, it's really promoted. But what we found interesting is that when we take these cells and we stimulate them with leucine, then that level of autophagy is regulated, or BCAT C induced autophagy can be dampened down. When I relate this then to amyloid, we can see that as the level of BCAT increases, the amount of accumulation of A beta in the cell increases, which is not good for the cell. However, with time, it indicates that as autophagy increases, the level of A beta decreases. So there is a correlation between the level of BCAT protein in the cell and how it regulates A beta. So the last part is whereby we take these cells and we incubate them with leucine. And as you can see, incubation with leucine really causes a significant decrease in the level of A beta in the cell. Now, the mechanisms underpinning this interaction are not clear to us right now, and we're currently looking into that in our laboratory. But our take-home points is that BCAT can control autophagy and levels of amyloid in the cell. Importantly, dietary leucine can redirect BCAT C involved autophagy if it's going awry, i.e. if it becomes dysregulated under disease conditions, and that may be important. But overall, I think the key thing to consider is that there are dietary components that can regulate these and fine-tune these mechanisms in the human brain. And if we can understand a bit more about them, that will be fantastic from a therapy-related approach. So I would like to thank the AIR UK for listening to me today, and for all of you for your participation.
Wonderful. Thank you very much, Myra, and thank you for sticking with us despite the challenging technical issues you've had.
I do apologise. There seems to be a problem with the internet at the moment.
That's all right. If we can get Jody back up, then we can move into our Q&A session. I don't know, Myra, if you wanted to try and disconnect and reconnect. We can and give a question for Jody to begin with, if that will help. So welcome back, Jody. So yeah, I've got a first question that's come through for you, Jody, while you were talking. And someone's asking, if you cut APP in the middle, might the pieces that result still clump?
That's an interesting question. I think I alluded to the fact that this is a promising alternative because it does cut in the middle of the protein. Certainly, if you recapitulate that in transgenic mice, then that leads to clearing of those plaques, and it can lead to reduced sort of cognitive decline. So I think it is a promising target, and the evidence seems to suggest that the fragments that are produced and the particular position where it cuts doesn't lead to sort of sticky bits of protein that are problematic.
Wonderful. And I think a question to kind of follow on from that was, say a drug that was able to be used to do this sort of activity, to cut it up to stop the clumping. I think a lot of the questions we get around potential treatments for dementia and for Alzheimer's is, when might those be given? So at the moment, at the point of people diagnosed, are already quite advanced.
Yeah.
So is the idea that you would be trying to pick up people earlier, and that's when these drugs might work?
Yeah, absolutely. And that's what I was sort of alluding to in the last slide there, the fact that by the time I think the symptoms start to present, the pathology is quite far advanced. So coupling that up with early diagnostics, early biomarkers could really help with that. And certainly if you had an instance where you could detect maybe through A beta oligomers, for example, these very short soluble forms of A beta, if you could detect those and you had a drug and you knew that it worked, but getting it in very late or getting it in when symptoms presented was having less of an effect, that really helps to jump in early and stop the decline, and then, you know, hopefully prevent any cognitive decline going forward.
And kind of sticking with that, we've got another question here that says, do beta amyloid plaques or the tau tangles just aggregate in the brain and neurons, or do they migrate into the blood? If so, could this be a target for biomarkers?
So they can, yeah, so they absolutely can go into the blood, and I think that's again, that comes back to that previous question, you know, of early diagnostics. So I mean, blood is a handy thing to get a sample from. It's a lot easier than sort of going into the brain. So if you can get blood samples or saliva samples, for example, that's a very nice way to be able to collect the samples and then diagnose in a sort of minimally invasive way. Where it gets a bit more tricky is what actually is it that you're detecting? And one of the problems with this, you know, I talked about a low as Alzheimer's seeing these amyloid plaques, these insoluble deposits, but we know actually that the correlation between those deposits and the severity of Alzheimer's disease is not great. And the current thinking is that it's these, as I said a few times, I think it's these very small soluble types of A beta that's problematic. And you start out with something that is one molecule, we call it a monomer, it then dimerizes, it can form trimers and tetramers, and you can not only get different oligomeric states, but you can also get different conformers. So you could have a dimer that has one structure and a dimer that has another structure. And it's one of the things that scientists are trying to do is to actually pinpoint the precise culprit, if you like, that is responsible. And I think if you could pick that out in the blood, for example, then that would be key. So not just seeing necessarily beta amyloid, but seeing the right types of oligomer as well.
Wonderful, thank you for explaining that. So we've got a question, I think, for you, Myra, and I think because we had a few technical issues, I think we just need to probably go over one thing that you were talking about, which was leucine. So Caroline's asking, is it a good or a bad thing to have in one's diet?
Well, basically leucine is an amino acid, so it's going to be present in all forms of protein. So we'll say, for example, if you ate a sandwich today and it had a high content of protein, once that protein is broken down, so one of the components of that will be leucine. So leucine is really important from the point of view as a nutrient signal. So it promotes protein synthesis. That's under normal physiological conditions. So it's a good thing. You need these amino acids and essential amino acids in the body. Now, with respect to regulating autophagy, we know that in response to leucine, autophagy is decreased or shut off, and protein synthesis is stimulated. And that again is just normal physiological response. However, in Alzheimer's disease, what we don't understand and what is not clear is how these two processes are controlled or regulated. Now, if there is a dysregulation of the autophagic pathway, some researchers believe that turning on that pathway would be, a very positive response. And there have been trials where they've used components or therapy molecules such as rapamycin. They've also used metformin even. All of these elements these compounds can stimulate autophagy. However, what is not known is that what happens when you get too much autophagy. If you put the pathway the whole other way, then do you start creating a situation whereby there's too much autophagosome synthesis and not enough clearance? So the problem is, Caroline, I think her name was mentioned that. So the problem is that it's the regulation that is what is key. So if we can regulate it through a dietary component such as leucine and know what that balance is, then that's where that's going to be really important. But as of yet, that balance and how that's triggered is not known. But we do know there is a relationship.
So kind of coming off the back of that question, are there studies going on in people, so outside of the lab, to look at how changes in diet or dietary supplements could try and alter this balance and shift it in the right way?
Yeah, there have been trials, and there are trials ongoing with numerous different permutations. Of course, most people would have heard Mediterranean diet, and also a diet where it is low calorie diet. So kind of what I was talking about, fasting diets, diets which are low in dairy products, that kind of thing. But the main, I think, drive from people in the field is, yes, a Mediterranean diet does seem to have positive associations. But as of yet, the trial design needs to be improved to really see that impact. Because if the trial is only for 6 months, for example, you can't really see the positive impact of a Mediterranean diet. However, if you put it on for a number of years, you may very well see the importance of following that type of diet. But there's also, of course, supplements, isn't there? That's big in the research arena at the moment. So vitamin D, for example, your B vitamins, folic acid, all of these vitamins are certainly big in the research field as to say whether they are a positive impact. Now, these vitamins will help your proteins or your enzymes operate maximally or more effectively in the body. So they can be a very good thing. But of course, you should always only take supplements and, you know, those equivalents with your doctor's approval, you know, so from that context, you know, be sure you're doing that. So yes, there are trials at this moment in time which are considering the use of vitamins and different diets in trying to, I think, delay disease onset would be a good way of putting it on.
So you mentioned there about the Mediterranean diet, and I think Belinda's been asking for any dietary advice, please. So what sorts of foods to include? So could you just tell us a little bit more about do you know the key components of
yes,
the
I do.
Mediterranean diet?
Yeah, absolutely. So it's not an actual fact. I think it's worth pointing out, it's not just the diet. I think I've frozen again, but I think you can still hear me, so that's okay.
Yeah.
It's not diet, it's also physical activity. So it's a combination of diet and activity seems to promote the best effects from a cognitive perspective. So like, so healthy foods. So we'll say, for example, all like your greens, for example, you know, everything from broccoli to spinach, those sort of things are really important. Olive oil, example, fresh fruit, nuts, legumes.
Or Myra's dropped off. Maybe I can pick up there. I think she covered most of the things. Oh no, she's back. Or is she?
Am I back? Okay.
You're back.
I think what thanks, Jody. So those are the elements that are really key to the Mediterranean diet. But again, I think it's a lifestyle change that's absolutely fundamentally important. And of course, there's a link between diabetes and your increased risk of developing Alzheimer's disease, which of course is related.
Yeah, so I mean, other things are obviously cutting down on alcohol, not smoking, and then, you know, there's anecdotal evidence around keeping your brain active as well, doing crossword puzzles, doing Sudoku puzzles, going for lots of long walks. All these kind of things, you know, are going to be helpful.
Wonderful, thank you, Jody, for stepping in there. Coming back to a question for you, I think, and so you mentioned in your talk some of the mutations in APP that you can find in genetic cases of Alzheimer's disease. And so we've had somebody ask, my mother passed away with Alzheimer's, no one else in the family has ever had Alzheimer's, does this make me more susceptible to the disease? So are there other genetic factors involved as well as those genes that you mentioned?
So as I think I said, the bulk of the cases are sporadic, which pretty much means that there are no underlying sort of genetic conditions that would predispose you to it. I would say it's most likely that if your mother has passed away from Alzheimer's disease, that would have been brought to your attention at that point, particularly if it had been diagnosed very early. So I think if somebody were to be diagnosed with Alzheimer's disease in their 30s or 40s because they'd come presenting sort of cognitive problems, then there could be a possibility, and it would be potentially be worth being checked. But for people that are sort of 60, 70, it's much less likely to be something where you need to be concerned about inheriting anything.
Wonderful, thank you. And kind of sticking with the risk theme that we've just had, Sandra's asked, so how does all of this relate to people from Black or minority ethnic communities? What makes them more susceptible than others? And I don't know, I don't think there's a full answer to it, but I wonder if you can talk about what we might know at this stage and what we need to find out about this.
I think the picture there is very unclear. I think there's a lot more work that needs to be done. Looking at particular cohorts, we know, as Myra sort of alluded to, that diet can play a huge role, and Mediterranean diet. We know Japanese diet, for example, can also be very helpful, perhaps things like lots of oily fish and things like that. But the honest answer is we don't really know. I think more work needs to be done, and the best thing that you can do is lead a clean and healthy lifestyle.
I think what I heard a good analogy is that what's good for the heart is good for the head. So taking care of your cardiovascular system, I do believe there's probably a link between that. And I think more recently in the field, there's a huge interest in the gut microbiome and the gut-brain axis. And as I mentioned in my talk, it's the importance of understanding that your health of your diet is very much related to your brain health. And I think keeping with diets that are good for the heart are really, I think, a positive way forward.
Yeah, I think with the minority ethnic communities, it's that there are risks with other conditions which are linked with dementia that they have increased risk of. There's probably some social factors there as well around not seeking a diagnosis or not having the same access to kind of medicine in the same way. So I think there is a number of questions that researchers are trying to answer around this, and I think we'll see more kind of shared around this as we get answers from those studies. So I think I'm just going to search for my final question. Actually, I think, Jody, I'm going to come to you on this one, because you talked quite a lot about amyloid, and people in the audience might be aware of particular drugs that have been trialed in recent years that have tried to clear amyloid from the brain. Why do you think these haven't shown any benefit in these studies so far?
So that's a really good question, and I don't know who asked that question, but they're right. They're right. There have been an awful lot of drug trials. There've been a lot of different types of drug that have been tried. Things like small molecules. My interests are, as I said, around peptides and peptidomimetics. There's a lot of work around antibodies. You know that we know antibodies are important. The important thing here is that the drugs have to be able to neutralize these most soluble, diffusible forms of A-beta that confer the cytotoxicity. And we know that earlier drugs have failed to do this. And also the other thing is, and that's something that I think we're more aware of now, so we can start to really gear our drugs at those particular types of amyloid. And the other thing is that dose is really important as well. So we have to have high exposures of the drug over prolonged periods of time. And I think that these two factors are really key to sort of clinical success.
Wonderful, thank you. Well, with us about 5 minutes to midday, there's just a couple of things I need to do towards the end. So I just want to take this moment to say thank you so much, Jody and Myra, for your really interesting talks, for answering all our questions. It was wonderful to have you.
Thank you.
Wonderful. So if our audience can stick with us just a couple minutes longer, because we've got a couple of poll questions we'd love you to answer to give us some insight into how you found today. So hopefully those will pop up on your screen now. And it's asking whether you would recommend these events to friends and family, how much you feel you might have learnt today, and whether you plan to attend in future. So I'll just give you a couple of moments to answer those and nervously wait and hope that you found it a good experience today. So if we've got enough people having done the poll, I reckon we can show the answers to it, show the results. There we go. And it's pretty positive. That's great. So you would recommend them, you feel you've learned something, and you do plan on attending again in future. So that's really wonderful for us to hear as we continue to plan more of these, now looking into the autumn as well. We'll also be sending a feedback survey along with the video recording of this event sometime next week. So please do complete the survey when you receive it, as it really helps us to have any feedback that you can give us. And you can also watch the video recording and see the presentations again then if you would like. So the next event in our series will take place on the 17th of June at 1 p.m. and it features researchers from our Thames Valley Research Network. And they will be taking us behind the scenes in labs in Oxford and Reading, showcasing how they develop potential medicines for dementia, how they use things like stem cells and cutting edge technology to understand better what happens in the brain in dementia. So do come along in a few weeks' time for what is set to be another great event. And you can find out more and sign up on our website. And we've also popped the link in the chat just now. I know that there were some questions that we weren't able to answer today, and some where you might need more personalised information or guidance on where to turn. Our Dementia Research info line is there to help. They can answer questions and signpost to other sources of information and support. So please do get in touch with them. So as we close, I want to say a huge thank you for joining us today. I hope you found it interesting and useful and will join us again in future for another event. Thank you so much and goodbye.